Hello everyone, we are from Group 5 and we're going to talk about mRNA 1273 COVID-19 vaccine. Our group member are Muhammad Ziyad bin Ismail, I Nadira Azmi binti Azmi and Narshan anak lelaki Tevarajan.
Okay, a very good day to everyone. I'm Narshan Devarajan, A184290 is my matrix number, and today my team is going to present about mRNA 1273 adenovirus vaccine. Okay, for my part, I'm going to briefly introduce what is virus and the basic principle of the vaccine. Okay, let's move. For the first part, I'm going to introduce what is virus is. Okay, virus is an infectious agent of small size and simple composition that can multiply only in living cells of the host, such as animals, plants, or bacteria. And nowadays, the virus which created the global pandemic is coronavirus, which causes the disease COVID-19. Formerly, this disease was referred to as 2019 novel coronavirus or 2019 nCovid. Coronaviruses are a family of viruses that cause illness such as respiratory diseases or gastrointestinal diseases. Okay. Respiratory diseases can range from the common cold to more severe diseases such as Middle East Respiratory Syndrome, MERS-CoV, and severe acute respiratory syndrome, SARS-CoV. Okay, a novel coronavirus is a new strain that has not been identified in humans previously. Once scientists determined exactly what coronavirus it is, they gave it a name, as in the case of COVID-19, the virus causing it is SARS-CoV-2. Coronaviruses got their name from the way that they look under a microscope. The virus consists of core of genetic material surrounded by envelope with protein spikes. This gives the appearance of crown. The word corona means crown in Latin, by the way. Okay, coronaviruses are zoonotic, meaning that the viruses are transmitted between animals and human. Okay, next. For the a global coordinated effort is needed to stop the further spread of this severe acute respiratory syndrome, coronavirus 2, SARS, also known as SARS-CoV-2, emerged in late 2019. This promoted an international effort to accelerate development of a vaccine. Thus, we need an appropriate substance to stimulate the production of antibodies and provide immunity against this disease, which try to act as an antigen without inducing the disease. For this definition, nothing can replace the vaccine, of course. Hence, a vaccine is needed to combat this life-threatening disease that occurring nowadays. And today my team is going to present one of the vaccine that have been developed recently by the National Institute of Allergy and Infectious Disease, the Biomedical Advanced Research and Development Authority, and Moderna, which known as mRNA 1273 vaccine. Okay. This candidate vaccine, mRNA 1273, encodes the stabilized pre-fusion SARS-CoV-2 spike protein. Also, this candidate vaccine is a lipid nanoparticle encapsulated nucleoside modified messenger RNA mRNA based vaccine categorized under adenovirus vaccine encodes the SARS-CoV-2 spike in bracket S glycoprotein stabilized in its profusion conformation. The S glycoprotein, as I mentioned just now in the bracket one, mediates host cell attachment and is required for viral entry, where it is the primary vaccine target for many candidates SARS-CoV-2 vaccines. Okay, clinically they have conducted a phase in human phase 1 in a clinical trial in healthy adults to evaluate the safety and immunogenicity of mRNA 1273. So I'd like to end my part with a video regarding to the basic principle of the vaccine. That's all from me. Thank you.
It all started with isolation of the virus from an infected patient in China. The coronavirus was sequenced and its genetic information was uploaded to a public database. Working together, scientists at the NIH and Moderna identified the sequence for a key protein on the surface of the virus, called the spike protein, as a good vaccine candidate. The instructions for making the spike protein were then encoded into an instruction molecule called mRNA, which could be administered directly to patients as a vaccine. The vaccine is injected just like other vaccines. The mRNA is taken to immune cells, where it instructs cells to make copies of the spike protein, as if the cells had been infected by the coronavirus. Other immune cells, shown here as red and orange figures, are then able to learn about the spike protein and develop ways to protect the person if they ever come into contact with the actual coronavirus. What makes this approach different is that you don't need to make the virus itself to make a vaccine, a time-consuming and intensive process. Instead, you use the information from the virus and administer the information directly to the patient. Essentially, the patient makes their own vaccine. This cuts out the middleman. Using this approach, NIH and Moderna were able to go from sequence to a vaccine ready for human testing in record time, just 42 days.
Hello everyone, my name is Ain Nadira Azmi binti Azmi and I'm going to explain about the method and the result of the COVID-19 vaccine mRNA 1273. The method used in the study were dose acceleration, open-label trial including 45 healthy adults 18 to 55 years of age. They will receive 2 vaccinations within 28 days apart with mRNA 1273 in a dose of 25 microgram, 100 microgram or 250 microgram. There were 15 participants in each dose group. For the phase 1 results, after first vaccination, antibody responses were higher with higher dose. On day 29, enzyme-linked immunosorbent assay anti-S2P antibody GMT, 40,227 in the 25 microgram groups, 109,209 in the 100 microgram group, and 213,526 in the 250 microgram group. After second vaccination, the titers increased. Serum neutralizing activity was detected by 2 methods in all participants evaluated, with values generally similar to those in the upper half of the distribution of a panel of control convalescent serum specimens. Solicited adverse events that occurred in more than half the participants included fatigue, chills, headache, myalgia, and pain at the injection site. Systemic adverse events were more common after the second vaccination, particularly with the highest dose, and 3 participants, that is 21% in the 250 microgram dose group, reported 1 or more severe adverse events. The table shows the percentage of severity of the symptom. Majority of symptoms resolve within 2 days. Some persist as long as 5 days. In this study, the methods used were randomized, observer-blinded, placebo-controlled trial was conducted at 99 centers across the United States. Persons at high risk for SARS-CoV-2 infections or its complications were randomly assigned in a 1:1 ratio to receive 2 intramuscular injections of mRNA 1273, that is 100 microgram, or placebo 28 days apart. The primary endpoint was prevention of COVID-19 illness with onset at least 14 days after the second injection in participants who had not previously been infected with SARS-CoV-2. For the phase 3 results, 30,420 volunteers who were randomly assigned in a 1:1 ratio to receive either vaccine or placebo. More than 96% of participants received both injections, and 2.2% had evidence of SARS-CoV-2 infection at baseline. Symptomatic COVID-19 illness was confirmed in 185 participants in the placebo group and in 11 participants in the mRNA 1273. Vaccine efficacy was 94.1%. Efficacy was similar across key secondary analyses, including assessment 14 days after the first dose, analysis that included participants who had evidence of SARS-CoV-2 infection at baseline, and analysis in participants 65 years of age or older. Severe COVID-19 occurred in 30 participants with 1 fatality. All 30 were in the placebo group. Moderate transient reactogenicity after vaccination occurred more frequently in the mRNA 1273 group. Serious adverse events were rare, and the incidence was similar in the two groups.
Hello, and my name is Muhammad Ziyad bin Ismail with matrix number A167628. Now we'll present about the discussion regarding our topics about our vaccines producer, which is Moderna, using the RNA come from the glycoprotein, spike protein of the coronavirus itself. So now we talk about the phases in the vaccine development. Maybe my friends has explained about the first phase and the third phase of the vaccine development, and I want to recall back about the first phase regarding the vaccine testing safety and the dosage. After it passed the first phase, maybe the sample taken for the first phase, like almost 100 people, like for Moderna, where the 5 people from the hundreds will receive the true vaccines by the Moderna, and the another 95 got just a placebo. The second phase is about the how they try to expand the safety trials. Maybe the quantity of the samples increase. And the third phase regarding the large-scale efficiency test. Most of the vaccines nowadays in any countries actually has come to the third phase. Maybe a certain at the second phase, but certain country like Russia or China try to bring it to the limited phase where the vaccine actually not quite ready for being commercialized, but it being given to certain people. And this actually not recommended by the specialists because it is too risky. Why? Because the safety of the vaccine is still uncertain. For now, Moderna is still in the phase 3 in terms of COVID-19 vaccine development, which is still not prepared to be commercialized, because after it being approved, then it will be commercialized, except if there's something wrong, it will be abandoned. And countries that have authorized these company vaccines, the Moderna vaccines, is Israel at the 4th of January and the European Union at the 6th of January. Now, Moderna tried to push from the phase 3 to the phase where the certain or limited person will receive the vaccines, which is previously I said that it is not quite good and too risky, said by the specialists, because it's not, the safety of the vaccine is still uncertain. So they intend to submit for an emergency authorization EUA with the US FDA in the coming weeks, and Britain and other countries also have begun giving emergency authorization to the vaccine based on the preliminary evidence that they are safe and effective. So now we'll talk about the adverse side effect of the Moderna vaccine itself, where the majority of the adverse side effect events were mild or moderate in severity, and the solicited adverse were generally short in period. That's being tested after they receive results from the phase 3, and based on this, most of the adverse side effect come to the secondary injection, where the secondary injection will be like a month or 3 weeks after the first injection. The first injection results shows that there's a grade 3 severe events greater than or equal to 2% in frequency after the first dose, including injection site pain. Like basically, if you receive any injection, you will got certain pain at the site of the injection. But certain case, there will be a grade 3 severe events. That this is being reported by the Moderna itself. So the secondary injection, maybe there will be fatigue, myalgia, arthralgia, headache, pain, and erythema, which is redness at the injection site. So we come to the most important discussion, whether the Moderna vaccine is good or bad. So pros of the Moderna vaccine is people won't get infected by the COVID-19 virus by taking the Moderna vaccine. That's the first thing. The second thing is the Moderna vaccine is easier to be stored as it remains stable at minus 20 degrees Celsius for up to 6 months and can be kept in the standard fridge for up to a month. And the third part is the efficiency of the Moderna vaccine in combating the COVID-19 is up to 94.5%. So the consequence of the Moderna vaccine, the bad side of the Moderna vaccine is it still has adverse side effects and not suitable for being commercialized yet. Because the second point is the vaccine is still in phase 3. The result can be varied based on the sample taken for the future. Maybe there's side effect that we still doesn't know or not being recognized.